Well, thank you very much for the invitation, and really very much interested in listening to all the talks today. Thank you, Mathilde. This will be about the Exposome perspective on toxicology, but I hope you will see that there are many connections. I mean, that are much more than I would have expected, but I will try to highlight them now that, okay. Okay, so I will speak about toxicology and really the bad things. I usually have the bad news here, but just to remind you that the environment is not only bad things. Of course, there are bad things in the environment. You have industrial problems, you have bad food, you have air pollution, but also there are some really good things. And sometimes the environment is a solution with green areas and blue areas and social environment, etc. So let's just keep this in mind because let's not think. But from now on, it's going to be the bad news. I'm sorry for that. So. Does pollution matter? Yeah, I think pollution matters. This is a slide from WHO showing the determinants of death worldwide, and it's determinants, it's not causes. I mean, and it shows really that total pollution worldwide is probably one of the most important determinant of death, almost like smoking or a little bit higher than smoking. Of course, most of this is air pollution because a lot is known about air pollution, and there have been. It's relatively easy to make these correlation for air pollution, and a lot of this is actually in low and medium income countries. But yet, I mean, pollution matters, so we have to take care of this. And this is another slide to tell you that concerning chemical pollution, there are still a lot of unknowns. This is from the European Commission, from the EEA, actually the agency, the environmental agency. It shows a sort of iceberg where basically around 100,000 chemicals are more or less in trade in the European Union at a certain level. 500 we know a lot about. 10,000 and 20,000 we know a little bit about. Maybe they are genotoxic or some effect, but basically 70,000 we know basically nothing on these chemicals. So there's still a lot to know, and I'm not mentioning here the mixtures that these chemicals can do. So there are still a lot of unknowns. Well, it's maybe our fault, toxicologists, because on the 500 we still work on the same stuff. I have been working on dioxin for a long time, and we know a lot. Maybe we know enough, but it's a model system, so it's maybe our fault. Now we have to get involved in these other chemicals. So, because I will be talking about the exposome, I thought I would give you a sort of perspective of the exposome and the different maybe definitions and inputs from the different people in the exposome field. So this is Chris Wilde actually coined the term exposome. He's an epidemiologist from IARC at that time, and the reason is that, well, when he was thinking about cancer and the determinants of cancer, well, a lot was known about genetics one way or another, but actually all the rest, like diet, mentioned that, pollution, stress, behavior, etc. I mean, all the rest was like in single spots. And he thought that it would be interesting to integrate everything that is not genetic, basically, everything about exposure in a single concept. Maybe because he was thinking it was also an inevitable part of the of the exposure. So basically, all life environmental exposure from prenatal period onwards. This is a lot, of course. This is huge. Actually, in one of my talks in the US, I was it was the Oscar period, and I said it's like everything, everywhere, all at once. Basically, this is and all your life long. Okay, so this is the first definition, Chris Wilde, and the idea is. This is the complement of the genome. So if you understand both, it may be reasonable. But now, when you have this kind of concept, then you have all other people in the field that try to make it a little bit more, maybe precise, well, to have their own input, actually, in the concept. And Rappaport, actually very early on, I will get back to Rappaport, had this analytical vision. If you analyze the blood exposome, you will learn more about chemicals. You will learn a lot about the exposome. So analytical, it was mentioned, high resolution mass spectrometry, different kind of chromatography. This is going to help us. Gary Miller was also in the analytical field, but he's a toxicologist, basically, which is, this is one of the toxicologists in the exposome field. And he basically said that the exposome is not only about listing the exposure, but also having some idea of their biological effect. So he integrated some impact of the exposome in biology. And of course, there's this ecotoxicological vision where people have suggested that we should not only be involved in what's happening in the human, but also see what's happening in the ecosystem and the interactions between humans and the ecosystem. It's a sort of One Health, planetary health perspective, but it's called sometimes the eco-exposome. Well, then we have been involved also with Price et al., a number of scientists in this field to identify what we would call functional exposomics, because basically still the exposome for many people is about only the exposures. So we thought like there are genomics and functional genomics. We identified exposomics, listing the exposure, and functional exposomic, while having a better idea about the biological and toxicological implication of the exposome. And other people like Roel Vermeulen introduced the exposome in the number of omics that you have mentioned a little bit earlier, but you didn't mention exposomics in your slides. So basically Roel said, well, we have all these metabolomics, transcriptomics, epigenomics, let's add one more omic, which would be the exposomics, and really do the network science system, network science with this. And when I mentioned exposure, I mentioned chemicals, I mentioned physical exposure, noise, I mentioned biological exposure, but also psychological and social exposure, and we have insisted on that in a paper with Paolo Vinis from Imperial and myself. Basically, how this relates to toxicology, it was not obvious at the beginning. Many of the actually initial studies on the exposome were done by epidemiologists and analytical sciences and few toxicologists. So we tried to bring in what would toxicology and exposome bring together. And basically, it's really about exploring multiple stressors at the same time or during time, delineating long-term effect, because the exposome is from birth. I mean, this includes the lifetime. So a better understanding in toxicology, long-term effect, bridging environmental threats and human threats, that's something we could do, and also the implication in regulatory and public health. I'll get back to that a little bit later. And just to mention Rappaport and state a few things, he really identified already in 2010, what he called the blood exposome. And basically, he said this includes all the chemicals, basically, in blood, which is a lot. I mean, this includes xenobiotics, all these contaminants that you have, but also, of course, microbial compounds, dietary compounds, but also endogenous compounds. And I don't think this, I agree that we have to include all this, but in a way, most endogenous compounds are known as the metabolome. People, refer to those as the metabolome and not, the exposome. So I think it's better to keep the exposome, blood exposome, or what we call the internal component of the exposome that you could get, as mostly the xenobiotics and their metabolites that got into your blood, the organism, microbial compounds that got into your organism, and some of the dietary compounds that got into your organism. And what is done endogenously, let's keep the term, metabolome. Otherwise, too much confusion in the field. There's a lot of confusion sometimes in some of the terms that people are using. We are trying to better identify these and make some sense of it. This is the history, basically, of the exposome. Birth, 2005. This is Chris Wilde. You can see here. I don't know if I can show. Yeah, I can show. I can see here, Chris. So it's 18 now. That's in France the majority. So it's mature now. Actually, nothing happened for a few years. Chris Wilde published his paper. It was rejected in most, journals. And eventually he published in a journal that I don't remember the name of actually now. And then 5 years, nothing much happened. And then suddenly people in the United States, mostly Rappaport, and in Europe started to getting interested in that. And you had a number then of research projects, both in the United States, in Europe, and then worldwide, trying to understand the exposome. Many infrastructures, facilities that try to help in characterizing the exposome. Hercules in the States was one of them. Now we have in Europe a facility, infrastructure in Europe. And in France, we have France Exposome, which is a facility that can, provide service if you need to assay whatever you have. If you have samples, call them. I mean, they can do a lot for you. And help you in understanding. And we have a number of projects in Europe. We have the EHEN project. And now we are really really speaking to each other worldwide, and people in the States, people in Europe are trying to further their understanding of the exposome and maybe try to work together in a, I hope, what would be a big exposome program like we had the genome program, and why not? So why would a toxicologist like me work, study the exposome, be interested even in the exposome? I have been working for a number of time on a receptor called the aryl hydrocarbon receptor. I will get back to this a little bit later. The aryl hydrocarbon, it was known as the dioxin receptor and a receptor for polyaromatic hydrocarbons. So I studied how this receptor induced a number of genes that metabolize xenobiotics, and basically how this pathway was both detoxification pathway and elimination, but also responsible for some toxicity. So that was my interest. I was an infamous reductionist at that time, and like many people in experimental biology. And how a reductionist like me suddenly got interested in the exposome? Well, this is the 1990 picture, perception of the aryl hydrocarbon receptor here, dioxins, maybe xenobiotic metabolism detoxification. But this is 2022 perspective on the, after a lot of work on knockouts, on different studies, in vitro, in vivo, in human, etc. So now we know that it does not only bind those pollutants and xenobiotics, it actually binds a number of course, many more xenobiotics, but a number of endogenous substances. You mentioned kynurenine and tryptophan, that's a good example. A number of dietary compounds. I worked a lot on polyphenols binding actually to this receptor, and microbial compounds, including those that are metabolized by the microbiome of tryptophan and many others. And it has a number of different functions in the immune system, vascular, hepatocyte, of course, cell cycle. And as a phenome, I mean, it's really involved in a very wide variety of effects. One receptor, many ligands, many effects. So how does this happen? Actually, this year we have, I mean, a group in France has been able, and in China actually at the same time, have been able to identify the structure of the receptor. And well, we didn't learn a lot from this now, but now we were going to learn, because now we can do the structure in the presence of the different ligands. Because our hypothesis, and based on toxicological work, is that depending on the ligand, this receptor does not have the same structure and does not bind to the same gene. It's a receptor that binds to gene to induce the genes. It does not have the same genomic effects depending on the ligand. So that's how we can explain having one receptor having different functions. So when you see xenobiotic and endogenous, microbial, dietary, this is the definition of the internal exposome by Rappaport, actually. So I thought, well, maybe we should be involved in that and get interested in that. So even a reductionist, when he looks at his work, sometimes it opens a very large public health perspective in many respects. So what we and others are doing is, we are in the lab, we are toxicologists, bioinformaticians, etc., but we collaborate a lot with epidemiology, with people in the exposure science. Where does it come from? Food here, but air, water, etc. So a lot of epidemiology, of course. The impact of stressor combination, I think this is really important. And then toxicology will work to improve the causality. And then implications in public health. This is basically how it's happening now in the Exposome project. And there are different frameworks for us that we are trying to work with. I usually call it the AAA because it's AEP, ADME, AOP. Well, the first two, AEP is aggregated exposure pathway. Learning how these different exposure end up in the human being, and learning about the kinetics, the importance of exposure from food as compared to air, as compared to water, as compared to other skin, other sources. So this is really now people that are in this field try to aggregate all these exposure pathway, determining the internal dose and the kinetics. This is very classical toxicology. ADME is very known, absorption, metabolism, excretion. And then the adverse outcome pathway, which is shown here. This is a framework that we all work in now in toxicology. Basically, it's a framework where we are putting the different events in relation to each other. Receptor activation, something happening in the cell, something happening in the tissue, and then the outcome at the end. These are the different events. We call them adverse outcome pathway. But of course, it's not a linear. It's really usually a combination. So networks are really important in there, and that's where many people are involved in now. And in our lab, for example, we are creating adverse outcome pathway, but we are also trying to find actually a text mining tool based on AI also to link with the literature different chemicals with events in the adverse outcome pathway. For example, we all know now that bisphenol A is not so good, but people are substituting it with bisphenol S, bisphenol F, and all the alphabet, basically. And we have done this, and we have shown that even bisphenol S and bisphenol F have similar effect as bisphenol A based on mining the literature and trying to put a weight on the evidence that is there. So now, ingested xenobiotics, if I focus really on xenobiotics, are composed of environmental contaminants, food contact material. We mentioned that. We really focus on these in the lab, but food contact are really important. Drugs, natural toxin. We shouldn't forget that nature is not so nice all the time, but there are many natural toxins. Food additives, this you know better than I do. Veterinary processing contaminants, substances of abuse. It's a big mix. And when it enters the body, of course, it gets an interaction with the microbiota, and I will tell you a little bit more. It gets absorbed. Also, the microbial compounds get absorbed. Liver is mostly responsible for metabolism, but it's not only liver. It's usually, the idea is here to eliminate these compounds, and most of them are eliminated, luckily, but some induce toxicity. There is also elimination through the biliary excretion, but also some reuptake, and that's the picture of these. So what are the techniques that are used in the, this is actually a picture from that Gary Miller gave me. It's basically mostly now based on mass spectrometry, but in the same sample you can do metabolomics with a lot of liquid chromatography and that is shown here, and mass spectrometry. But if you do gas chromatography, you have a better chance of getting hydrophobic compounds. And many of these xenobiotics are, in fact, hydrophobic. So with the same sample, you can get hundreds and thousands of chemicals that are mostly contaminants, xenobiotics, and at the same time, the metabolic. And if you compare both, you can have some idea about possibly the effect. Although there's a time problem here, but at least you can have both the metabolome and the exposome in basically with a single sample. So there are many effects, actually, and now exactly like in cancer, for example, you have the hallmarks of cancer. We in environmental health have the hallmark of environmental health, and I'm going to give you some example of each. Of course, what has been mostly studied over the years is genomic alteration mutations, genotoxicity. For example, smoking, aflatoxin was mentioned a little bit earlier from food. Epigenetic alteration, many endocrine disruptors induce epigenetic changes in methylation, although it's not always very easy to make a correlation between what's happening and these genes, the methylation, and the final impact. In some cases, it makes biological sense, so this is good, but it's not always easy to do that. Mitochondrial dysfunction is a major issue, and we can test that, and many pesticides have this kind of effect. Of course, endocrine dysfunction, I will tell you more about it. Altered intercellular communications, we have done a lot of work on dioxins and how they alter the communication and how they induce migration of cells. And this is correlated with clinical study that we have carried out in breast cancer, showing that there is a correlation between the dioxins that are present in the breast fat tissue, we assay it there, and the capacity of breast cancer to induce metastasis, to progress, cancer progression. So it makes sense biologically, and I think this is a very important. Of course, interaction with the microbiome, nervous system, oxidative stress, that's a major impact. And of course, immunotoxicity. The example here is perfluorinated compound, the eternal substances you know more about them in the US than actually in France, but it is starting now. These are really bad compounds and have a lot of immune toxic effect and other effects too. Concerning endocrine disruptor, it's really one of the major fields where we, in our field, in the exposome and toxicology, we work on. It's really an exogenous substances that alter the function of endocrine system and consequently cause adverse effect. The most difficult part is consequently, showing that it's by inducing the hormonal disturbance that this is leading to a toxic effect. That's what takes more time to us. And interestingly, it started in the ecosystem. It didn't start in human studies. In the ecosystem, in contaminated lakes, people have been looking at alligators and the reproductive capacity of alligators in contaminated lakes, and that's how they started to make the correlation between reproductive system, hormonal system, and contamination. That's really how it started, but then, of course, now you have a number of substances. I'm not going to cite them all. A number of substances, many pesticides. So that should bring many plasticizer that contaminate food. So I mean, it's a huge bunch. But I want to be to be careful here because most of these are not bona fide endocrine disruptor. Most of them are putative endocrine disruptor. Why? Because we don't have the complete demonstration that it is by modifying the hormonal system that they cause the toxicity. We still need some of the links here. So there are, I would say, maybe tens of endocrine disruptors, but maybe thousands of putative endocrine disruptors, so we have to be careful now. Of course, endocrine disruptors are present everywhere, including in food, so I'm going to tell. Now, concerning the time issue is a major problem for us in toxicology. The exposome says it's long period. It's your exposure over the lifetime. So how can we model this? I mean, and there are different models. This is an attempt. Either when you need to have a toxic effect when the exposure is repeated, like smoking, for example. Or persistence, dioxin. If you get dioxin now or perfluorinated compounds, it will remain for years in your body, stored somewhere and then released at a low level. So that's another. But maybe the most exciting one is the programming, changes in the programming in this early developmental period. Some people call it the DOHAD concept. And the exposure during the fetal period or early childhood increases the risk of having some kind of toxicity much later in life. And the mechanism here is likely to be epigenetics. So I'm going to skip this. Epigenetics is, I think, very important for all of us, I guess. There are some examples in toxicology where this is tributyl tin, where the effect, for example, on the adipose tissue and obesity later is correlated with some epigenetic marks that make sense, that where the genes that are induced through these epigenetic effects actually are involved in having a bigger storage for fat. So it makes sense. So epigenetic can be part of the mechanism, which is great. But even if it's not part of the mechanism, it actually can be used as a biomarker. We may need biomarkers that tell us what will happen later in life. That's maybe the most difficult part that we have. And we are all looking for biomarkers of effects. Then another issue that is really important in our field and in exposome, in food toxicology is the mixture. We are always exposed to mixtures and not to a couple of compounds, but actually to hundreds and thousands all the time. So here I just will mention two research objectives. One is to identify co-exposure. I mean, when a group, for example, children, what are they exposed to at the same time? I mean, that's a question that one can ask. The other approach is to determine what is the impact of a determined mixture of pollutants. So they are related, but it's different. Mostly the analytical people are working here, and mostly the toxicologists are in the second. So these are the two issues. And in a regulatory perspective, this is absolutely critical, because if you work on each endocrine disruptor alone, you may have all these below the threshold, the sanitary threshold, and they are okay. You can, consume your apple or whatever you want. But if you put them together, especially they are mixed together, especially if they have the same mode of action, that's where toxicology is important. If they have the same mode of action, it makes perfect sense biologically to add the concentrations of these compounds. So, and if you do it, you may get above the threshold. And nowadays, in the regulatory field, each substance is studied alone per se. It's not, so we really need to do something about it because we are not really protecting the population. There's one exception, dioxins. For 30 years, when you say you have that much of dioxin, in fact, people are just taking 30 compounds that are similar to dioxin and adding them, making these additions, and you will have toxicology equivalent to dioxin, and that's done. I don't know why it's not done in the other. You can guess, probably. Another important issue that we have done, actually this is in the lab, is the interaction between dietary imbalance and chemicals. I think this is a very promising field. There are many examples. This is one example on the liver in rodents. This is experimental, where we have studied dioxin on one hand and high-fat diet on the other hand, and then assayed for fibrosis and fat accumulation. And basically, when you have both together, you have more fibrosis and more fat accumulation. Maybe synergistic, I don't know. It's not precise enough to say, but at least we should know that there is a great interaction between exposure to chemicals and dietary imbalance. So a new field, actually, is microbiome. And of course, it is really concerning us a lot. This is just a summary of the different interactions between the microbiome and xenobiotics. They can accumulate in the microbiome. They can be metabolized. So what you get is the metabolite from the microbiome and not the initial xenobiotic. They can change themselves the profile of the microbiome or induce growth, or inhibit growth. So there are many examples. And in the field of, for example, metformin acts on the microbiota, and this is a positive way, in a rather positive way. It may be part of its drug effect. However, glyphosate, and I'm sure you know about glyphosate, the pesticide, so it has actually, it induces an imbalance in the microbiome, and it is maybe part of its toxicity. So both ways, xenobiotic can change the microbiome profile. There is a very famous example, I don't know if you're aware about melamine. Melamine was added to milk and things just to increase the amount of nitrogen, so that people think, they dilute the milk and they add melamine, so people think that they have the right amount of proteins because of melamine. And it has been done, and basically it's extremely toxic to the kidney. And people didn't know the mechanism for a long time. And the mechanism is here. This is metabolism of melamine in the microbiome, and it leads to cyanuric acid, which is a contaminant to the kidney. So now we know that the microbiome is responsible for this toxicity of melamine. Back to the AHR, the aryl hydrocarbon receptor, as in barrier system in the intestine, it has a number actually of effect, mostly including on the stem cells of the colonic part, including on different islets of lymphocyte and lymphocytic tissue. It's really important in immunology, this receptor. So it has a number of effects, and actually maintaining more or less the barrier integrity. But another picture here of this receptor, my favorite receptor, the AHR. As I said, there are endogenous compounds, xenobiotics, microbial compounds, dietary compounds that can bind with a number of different functions. And I think when you mentioned these metabolites of tryptophan, some of them are very good AH receptor agonists, extremely well. Quinoridine is also, but it's a weak agonist. The others, indole, derived from indole, are extremely good agonists. So I wonder whether this could be, one of the mechanisms. We cannot put all the mechanisms on this receptor, but it is really important in the gut and in microbial identity. So just a few conclusions here. One of them is, I didn't speak, well, we have 100,000 chemicals that are, in production, in industrial chemicals, etc. And in fact, we have only dozens of enzymes and receptors that are in charge of detoxification. So what's the secret? The secret is in the flexibility of the structure and maybe the multifunction of these enzymes. So that's a major issue that we have to deal with to understand it better. So we have hundreds, sometimes thousands of ligands for a single receptor or a single enzyme. Some cytochrome P450 bind a number of different ligands. This lack of specificity is such that there is a cost to this adaptive pathway. There is a cost. There is competition sometimes, inefficient reaction, release of reactive oxygen species. This is why these pathways, which are in the detoxification and elimination, in fact, are also in the long run also responsible for some chronic toxicity. So we have to understand this better. So people who tell you, it doesn't matter, you can take all these xenobiotics because you have the system to get rid of them. No, the system is not perfect. Okay, as I said, these can share the same pathway, and now the microbiome is involved in the fate of xenobiotics, which was not recognized actually early on. Okay, so why are we interested also in exposome? We need a better integration between toxicology, epidemiology, exposure sciences, bioinformatics, modeling, you name it, nutrition, of course. It can help us link to exposure. And if you want to do risk assessment, you don't only want to do what is the effect, but you need where the exposure is coming from. What should the government do? And a better understanding of the mechanisms of stresses. I think this is what mentioned earlier, and I think we can do something about it. The problem of mixture is on the top list. The vulnerability of certain period, better understanding this vulnerability, I think it's exactly the same in nutrition as it is in toxicology, and of course the long-term effect that I mentioned. So there are a number of regulatory implications. I mentioned mixture, but there are many, many others, taking into account, for example, some outcomes that were not taken into account in regulatory agencies. I mean, recently bisphenol A, the safe level of bisphenol A has been decreased 20,000-fold by the EFSA, the European agency, because suddenly they took immunotoxicity into account, which means that initially we were all below the threshold. Fantastic. But now we are all above. There are also, I think it's really interesting to take into account the exposome and multiple exposure, because I think public health messages should be coherent. You cannot say at the same time that pesticides are not good and that you should eat fruits and vegetables that most of them have pesticides. I mean, so. And also you mentioned precision nutrition and precision prevention in general, and we can do a lot in the exposome world, first by assaying, second by having these wearable devices that can tell you what are you exposed. I mean, a number of issues. Now, I think this should go along the same line. And finally, and this is for you, Sal, finally, a toxic score or an eco score or planet score, a score that tells everybody that there is something maybe not so good in what they are buying. It's much more difficult. The government every now and then says, we have to start working on it. And then I don't hear about them. 1 month later, nothing happens. And they change the minister, and then this goes on for a while. So maybe this is something we should talk about. Thank you very much.